For decades, the standard explanation for Neanderthal extinction pointed at us, Homo sapiens, outcompeted them into oblivion. But between 2014 and 2024, ancient DNA from sites spanning Spain to Siberia has rewritten that story. The Altai Neanderthal genome, sequenced at 52-fold coverage, contained over 100 megabases of runs of homozygosity, the genetic signature of parents who were half-siblings. Species-wide effective population was roughly 3,000. Neanderthals were not beaten by a superior rival. The DNA says they were already doomed, and the evidence for why starts with a single genome pulled from a Siberian cave. In 2014, Svante Prüfer and his team at the Max Planck Institute unveiled a genetic record that would upend assumptions about Neanderthal life. From a single toe bone, unearthed in the icy layers of Denisova Cave in Siberia, they extracted DNA of astonishing quality, sequenced at 52-fold coverage, a level of detail never before achieved for ancient hominins. This was not just a technical milestone, it was a window into a vanished world, and what it revealed was both extraordinary and unsettling. The Altai Neanderthal's genome was riddled with over 100 megabases of long runs of homozygosity, stretches where both copies of DNA are identical, inherited from the same ancestor. Each segment exceeded 10 megabases, a length almost unheard of in modern populations. In today's world, such patterns are seen only in the most isolated human groups, and even then, rarely at this scale. The genetic signature pointed to parents who were half-siblings or perhaps double first cousins, suggesting a level of inbreeding that would raise alarms in any population geneticist's mind. Prüfer's group ran computational models to test the limits. Could this be the result of a single generation of close-kin mating? The answer was clear. The pattern could only be explained by repeated inbreeding, generation after generation, within a group so small that genetic diversity was collapsing. The effective population size, calculated from the genome's heterozygosity, hovered around 3,000, an order of magnitude lower than estimates for contemporary Homo sapiens in Africa or Eurasia, where effective sizes ranged from 20 to 30,000. The Altai genome also showed a marked deficit of rare genetic variants, and the site frequency spectrum was skewed, both hallmarks of a population that had endured a long-term bottleneck. The evidence was overwhelming. This was not a temporary setback, but a chronic state of isolation. Prüfer's team took extraordinary care to rule out contamination, keeping it below 1%, and filtered out the characteristic damage that plagues ancient DNA. The Altai data set a new gold standard for the field, but its implications were even more significant. The findings forced a fundamental rethinking of Neanderthal demography. Rather than a robust, widespread species, the genetic evidence painted a picture of a population teetering on the edge, fragmented, isolated, and shrinking. Yet, this was only a single genome, a lone voice from the past. Was the Altai Neanderthal an outlier, the product of a uniquely desperate community? Or did her DNA reveal a wider pattern of collapse? The urgency was unmistakable. To know if inbreeding defined Neanderthals everywhere, researchers would need more genomes, more data, and new methods. For now, the Altai specimen stood as a stark warning, a genetic red flag waving from the depths of prehistory. In 2021, Harold Ringbauer at the Max Planck Institute introduced a hidden Markov model that changed how researchers detect inbreeding in ancient DNA. This method, built for the unique challenges of degraded Neanderthal samples, treats each 100 kilobase window as either autozygous or not, using genotype likelihoods instead of raw calls. The model tolerates sequencing errors by allowing a minimal heterozygosity rate within runs of homozygosity, and it skips windows with more than 30% missing data. Before analysis, the pipeline strips out postmortem damage, especially C to T and G to A transitions at read ends, and it systematically removes samples with contamination above 3%. Ringbauer's team validated the approach, using 2,500 simulated pedigrees and 1,000 modern genomes with known family relationships. The algorithm's inferred runs of homozygosity, length distributions, matched actual kinship coefficients with a Pearson correlation of 0.96. This level of accuracy proved that even genomes sequenced at just 3-fold coverage could reliably reveal long runs of homozygosity. The method's reliability was confirmed across 3 length bins: short tracks from 0.5 to 2 megabases, intermediate between 2 and 5 megabases, and long tracks above 5 megabases. In Neanderthals, tracks over 5 megabases directly signal recent parental relatedness, such as half-sibling or cousin unions. This hidden Markov model made it possible to analyze dozens of low-coverage Neanderthal genomes from across Eurasia, not just the rare high-coverage outliers. Simulations showed that long runs of homozygosity, the hallmark of close inbreeding, remained detectable even in sparse, fragmented DNA. The algorithm's tolerance for error and missing data means that results from Altai, Chagyrskaya, Vindija, and Mesmaiskaya can be compared on the same quantitative footing. By standardizing runs of homozygosity detection, Ringbauer's method transformed what had been a surprising finding in one Siberian specimen into a systematic tool for reconstructing Neanderthal demography across time and space. The scientific community now had a way to measure inbreeding and effective population size with confidence, regardless of sample quality. This methodological breakthrough underpins every subsequent claim about Neanderthal genetic collapse and allows the field to move from isolated case studies to broad, population-level inferences. In 2022, a team led by Fabrizio Maffesoni and Loritz Skoff at the Max Planck Institute published a genetic analysis of 13 Neanderthal individuals from Chagyrskaya cave in Siberia. Sequencing revealed a direct father-daughter pair among the remains, confirmed by autosomal overlap and identical Y chromosome haplotypes. Kinship coefficients placed their relationship at 0.25, matching the expected value for a first-degree connection. This is the first nuclear DNA-based pedigree identified in a Neanderthal group. The same study uncovered additional lateral relatives, including likely uncle-niece and cousin pairs, all dating to a single generational window between 54,000 and 58,000 years ago. Pairwise, genetic dissimilarity across the 13 individuals allowed the team to estimate the local census size. Simulations of a closed, randomly mating group produced the observed pattern only when the community comprised between 10 and 20 adults. This figure stands in stark contrast to the effective population sizes of contemporary Homo sapiens, which ranged from 10,000 to 20,000. The Chagyrskaya group functioned more like a small village than a tribe, with every member genetically close to every other. Analysis of mitochondrial and Y chromosome diversity provided insight into the social structure. Mitochondrial lineages were 2.5 times more diverse than Y chromosome lineages, indicating that females moved between groups while males remained in their birthplace. Despite this female-mediated gene flow, the genome still showed extensive runs of homozygosity, comparable to those seen in the Altai and Vindija Neanderthals. The presence of long runs of homozygosity in multiple individuals confirmed that inbreeding was a persistent feature, not a rare event. The Chagyrskaya findings demonstrate that extreme demographic contraction and close kin mating were not unique to the Altai Neanderthal. Instead, these patterns defined Neanderthal communities across Eurasia. The evidence from Chagyrskaya translates genetic signals into a lived reality. Neanderthals often lived in micropopulations where nearly everyone was family, and the genetic consequences of such isolation were severe and ongoing. Petrilocality, a system in which males remain in their natal group while females move between communities, left a distinct genetic footprint in Neanderthal populations. Analysis of genomes from Chagyrskaya cave and other sites shows that mitochondrial DNA diversity was 2.5 times higher than Y chromosome diversity. This imbalance signals that while women occasionally migrated between bands, male lineages were largely static. As a result, the effective population size for Neanderthal males underwent a collapse to a fraction of the total census. The Y chromosome bottleneck, confirmed across L. Cedron, Vindija, and Spy, reveals that a single paternal lineage persisted across Western Eurasia for tens of thousands of years. With so few breeding males, every new generation was forced to draw mates from a shrinking pool of close relatives. The outcome was intensified runs of homozygosity, especially in the long tracts exceeding 5 megabases that indicate recent consanguinity. Even with incoming females, the limited number of unrelated males meant that inbreeding could not be avoided. Computational models by Ringbauer and colleagues show that this social structure slashed the effective population size well below the census, amplifying genetic drift and the fixation of harmful mutations. In such an environment, even moderate demographic shocks would have catastrophic genetic consequences. The persistence of petrilocality, combined with geographic isolation, transformed each Neanderthal band into a demographic island. Reduced female gene flow could not compensate for the loss of male diversity, making each group more vulnerable to stochastic extinction. The pattern holds from Siberia to Iberia, linking the microscale kinship networks observed at Chagyrskaya to the continent-wide collapse of Neanderthal lineage diversity. In 2024, Ludovic Slimak and his team published the genome of the Thorin individual from Grotte Mandrin in southern France. Thorin's DNA revealed a striking pattern. For at least 50,000 years, this Western European lineage remained genetically sealed off from both Eastern and Central Neanderthal populations. Analysis of autosomal divergence placed the split between Thorin's ancestors and other Neanderthal groups at more than 50,000 years, a separation confirmed by the absence of shared recent ancestry with specimens from Vindia, Chagerskaya, or Mesmaiskaya. The divergence was measured using the autosomal clock, which tracks the accumulation of genetic differences over time. Thorin's genome carried unique combinations of derived alleles not found in any contemporaneous Neanderthal, supporting a scenario of long-term isolation. Slimak's group calculated that gene flow into this lineage effectively ceased after the initial split, with only negligible contact events inferred from the data. The Thorin genome's heterozygosity was among the lowest recorded for Neanderthals, mirroring the severe reduction observed in Altai and Chagerskaya individuals. The results indicate that by the time Homo sapiens entered Europe, Western Neanderthal populations had already experienced tens of thousands of years of demographic isolation. Thorin stands as direct evidence that the species' fragmentation was not a recent development, but a persistent condition stretching across the continent and deep into the past. The findings reinforce a central conclusion. Neanderthals were divided into small, isolated units for much of their existence, and the western edge of their range was no exception. Mitochondrial and Y chromosome data reveal that Neanderthal lineages were already collapsing long before their disappearance. At Mesmaiskaya Cave in the Caucasus, researchers sequenced mitochondrial genomes from two individuals separated by up to 25,000 years. Both carried nearly identical mitochondrial DNA, indicating minimal maternal turnover across millennia. This pattern, published by Pabo's group, stands in stark contrast to the rapid mitochondrial replacement seen in modern human populations. The Mesmaiskaya findings suggest a female effective population, size so low that new maternal lineages failed to establish, even over tens of thousands of years. On the paternal side, Mendez and colleagues in 2016 reconstructed the Neanderthal Y chromosome from 3 sites spanning Western Eurasia, El Sidron in Spain, Vindija in Croatia, and Spy in Belgium. All 3 males belong to a single Y chromosome haplogroup with no evidence of regional variation. This uniformity persisted across a geographic range of more than 2,500 kilometers and over at least 20,000 years. In modern humans, even small populations maintain dozens to hundreds of Y lineages. The Neanderthal pattern reflects a severe, long-term bottleneck in male ancestry. By the late Middle Paleolithic, both maternal and paternal lineages had stagnated. The loss of mitochondrial diversity at Mesmaiskaya and the continent-wide dominance of a single Y haplogroup demonstrate that Neanderthal populations were shrinking and isolated. These organellar bottlenecks predate the arrival of Homo sapiens in Europe and underscore the depth of genetic depletion within the species. Lee and colleagues in 2024 quantified gene flow between Homo sapiens and Neanderthals using genome-wide comparisons from over 20 ancient individuals. Their analysis found modern human DNA entered Neanderthal genomes at rates roughly double the reverse, yet the total introgressed fraction never exceeded 2% of the Neanderthal genome. Most Neanderthal groups received only sparse, patchy segments from Homo sapiens, and these fragments failed to spread widely across isolated bands. Directionality of gene flow was confirmed by tracing derived alleles unique to Homo sapiens, revealing that introgression events were infrequent and geographically restricted. Agent-based demographic models published by Vason in 2019 simulated Neanderthal population dynamics using census sizes and inbreeding coefficients derived from ancient DNA. These models excluded external pressures from modern humans, focusing only on internal factors, birth rates, death rates, and the genetic load from inbreeding. For bands of 20 individuals or fewer, the probability of extinction within 10,000 years exceeded 50%. When realistic inbreeding levels, as observed in the Altai and Chagerskaya genomes, were included, extinction risk rose above 75%. Even with occasional gene flow from modern humans, simulated Neanderthal populations could not achieve the tenfold increase in effective size needed to escape this demographic trap. The limited and asymmetric gene flow, coupled with persistent low effective population sizes, meant that Neanderthal groups remained isolated and genetically compromised. Modeling shows that the addition of 1 or 2% modern human DNA did not reverse the accumulation of harmful mutations or restore lost diversity. The final Neanderthal populations, fragmented and inbred, faced extinction as a mathematical certainty. Today, non-African humans carry 1 to 2% Neanderthal ancestry in their genomes, but the original Neanderthal Y chromosomes and mitochondrial lineages are entirely absent. The evidence from ancient DNA and demographic simulations closes the case. Genetic rescue never arrived, and the end of the Neanderthals was sealed long before their last contact with Homo sapiens. Genetic collapse, not conquest, remains the primary verdict from sequenced Neanderthal genomes published by Pabo's team at the Max Planck Institute. Today, over 40% of Neanderthal DNA persists, scattered across billions of modern human chromosomes. As demographic crises intensify in endangered species worldwide, the Neanderthal record serves as a warning. Genetic isolation can drive extinction long before any rival arrives. What happened once can, and does, happen again.